Today we're going to be looking at DNA. So let's put that as our heading for today, DNA. And this stands for deoxyribonucleic acid. That's deoxyribonucleic acid. And we want to look at this term in a bit more detail. So it's got something called nucleic acid within this name. So let's zoom in a little bit more in this term because it comes up again and again when we are talking about DNA. So nucleic acid. Well, what exactly is a nucleic acid? A nucleic acid is simply something which stores and processes genetic information. So stores and processes genetic information within a human cell. When we're talking about nucleic acids, we've actually got two types of nucleic acids within the human cell. And the first of these types, well, we've spoken about it already, it's going to be DNA. And the second of these nucleic acids, it's RNA, which I'm sure you will have heard of before. It's ribonucleic acid. Now, there's a slight difference between the structure and the function of DNA and RNA. RNA, ribonucleic acid, is something that we're going to look at in another video in a bit more detail. So, DNA, we've covered the nucleic acid part of the name, right? But we've still got this deoxyribo part of the name, which we're not sure exactly what this means. It actually stands for deoxyribose. So let's look at this in a bit more detail. We'll take a clear slide and we'll put another heading called DNA structure, as we're looking at the structure now in a bit more detail. Now, when we're talking about DNA, we're going to define it as a double-stranded helix. So that's a double-stranded helix. And that's basically just describing the structure of DNA and what it looks like. So if we draw a basic diagram of DNA, we'll actually see that it looks something like this. And we have these rungs in the middle of this twisted ladder-like shape. So it's got two strands, so it's double-stranded, and it's a helix because it's twisted in its shape. Now, DNA, it has three components. So it has three main components to it. And the first of these components is something called your pentose sugar. That's pentose sugar. Now, we'll remember that pent, whenever we see this term, it means 5. So this is a 5-carbon sugar. Now, the name of this pentose sugar, when we're talking about DNA, is actually deoxyribose. Yes, that's right, it's deoxyribose. Now things are starting to click. So this is where the deoxyribo part of the deoxyribonucleic acid comes from. This refers to the pentose sugar within DNA. So that's one component within DNA. Now, the second component is something called your phosphate group. That is your phosphate group. And this refers, in the context of DNA, is going to be your phosphoric acid. So phosphoric acid is your phosphate group within DNA. And the third component within DNA is something called a nitrogenous base. And there is 4 different bases within DNA. Now, these nitrogenous bases, they can actually be split up further based on their structure. So they can be subcategorized into purines and pyrimidines. So purines and pyrimidines. Now, we said that these are based on their structure, and purines are a double ring structure. So double ring, and pyrimidines are a single ring. And that's the difference between both of these. Now, there's 4 bases, so there's going to be 2 bases within each. So when we think about purines, what we have is something called adenine. and we'll put A for short, and we have guanine, and we'll put G for short. And when we think about pyrimidines, we've got cytosine, we'll put C, and we have thiamine. So cytosine and thiamine are our pyrimidines. Now, these 4 bases, what they do is they pair up within a DNA molecule, and they don't actually pair up within their subcategories based on structure. So they don't pair up according to whether they're purines or they're pyrimidines. A actually joins to T, so adenine joins to thymine, and guanine joins to cytosine, and vice versa. So these are always going to be found as pairs within a DNA molecule. So let's look at this structure in a bit more detail. Okay, so we'll flesh this out a little bit and draw a larger DNA molecule. So we have one strand here, and we have one strand cutting across like this. And this is a very basic diagram, but we're just drawing it to illustrate our point and to demonstrate the structure of DNA. So what you have is each strand has a backbone of 2 of these 3 components, and these are your pentyl sugar, so a deoxyribose, and your phosphoric acid, so your phosphate group. And this structure repeats all the way down the strand. And in the middle, what you have is you have a nitrogenous base, and this always comes off your deoxyribose. Okay, so each nitrogenous base is found within the middle of both strands, and this comes off your deoxyribose molecule. Now these are joined by weak hydrogen bonds. So in between you have weak hydrogen bonds joining both of these, which comes into play when DNA molecules are separating into single strands, for example, if the nucleus is dividing. So this is how these 3 components come together within a DNA molecule. But we want to also talk about another structure, which is the basic component of DNA, called a nucleotide. So let's put that as our next heading, a nucleotide. And this is a term that you might or might not have heard of, and it's something, again, that we want to define so it's clear in our minds exactly what it is in context to DNA. So what we can say is it's the smallest structural unit within DNA. So the smallest structural unit in DNA. Okay, so that gives us a brief framework of what we're working with. And again, to illustrate this, we should draw a diagram. So again, we've got a DNA molecule, and we've got one strand over here, and we have another strand over here, and again, just a very rough diagram which is just illustrating our point. So this is a part of a DNA molecule, and like we talked about, what we have is we have deoxyribose joined to phosphoric acid, joined to deoxyribose, joined to phosphoric acid, and so on and so forth. And again, we'll just draw this backbone structure alongside each strand, just so we can clearly see exactly what we're talking about. So alongside this, what we have is we have nitrogenous bases coming off our deoxyribose, right? And they only come off our deoxyribose molecule. And we'll just draw this at the bottom as well, just to illustrate our point. So we've got deoxyribose and we've got phosphoric acid, and so on and so forth. And then we have our nitrogenous bases coming off our deoxyribose or pentyl sugar. So let's say, for example, that this top deoxyribose here, the nitrogenous base is adenine, right? Now, where does a nucleotide come into all of this? Well, a nucleotide is simply composed of one pentose sugar, right? One pentose sugar. So that's going to be our deoxyribose plus one phosphate group, which is going to be our phosphoric acid plus one nitrogenous base. So that is actually what a nucleotide is. It's just one of each of the three components of your DNA molecule. So let's circle this, right? So we've got one deoxyribose here, one phosphoric acid here, and one nitrogenous base, which is adenine here. And there's actually 4 nucleotides which are based on which nitrogenous base is included within each one. So we've got adenine here. So the name of this nucleotide is going to be deoxyadenylic acid. Deoxyadenylic acid. And say, well, adenine is also going to be joined to thymine, isn't it? Because they always pair up. So you've got another nucleotide here. And the name of this is going to be deoxythymidilic acid. So that's deoxythymidilic acid. So that's our second nucleotide. And we've got another one here, which has cytosine within it. So we'll just circle this. This is our third nucleotide. And this is going to be called deoxycytidilic acid. Deoxycytidilic acid. And, well, this is going to be joined to G, isn't it? It's going to be joined to guanine. So there we have our fourth nucleotide. And this is going to be called deoxyguanilic acid. So that's your 4 different nucleotides. So let's label them. You have deoxyadenylic acid, you have deoxythymidilic acid, which has thymine, you have deoxycytidilic acid, which has cytosine as your nitrogenous base, and you have deoxyguanilic acid, which has guanine as your nitrogenous base. Crazy thing here is, right, that each DNA molecule actually has over 45 million nucleotides. Yep, that's over 45 million nucleotides. That's absolutely mind-boggling. So that's what a nucleotide is, and that's a quick overview of the structure and the function of DNA.